Hi, my name is Lily Hiltebeitel. I'll be talking about my thesis, which is investigating the evolution of non-coding regulatory elements and how they connect to human aging. So because of lack of time, just as a disclaimer, I'll be mostly looking at notes for this draft as I talk, but in future recordings, I'll definitely be more rehearsed and will probably sound more natural. So I'm sorry in advance for that. So here's a brief overview of what I'm going to be looking at in this presentation. And what is non-coding DNA, just for those who are not as familiar? So this figure gives a broad overview of the role of non-coding DNA in our genome. So as you can see, only about 1.5% of our genome is protein coding genes, which is what we typically think of when we think of our DNA. And the vast majority actually of our DNA is non-coding. So you might remember learning in high school or earlier courses that our non-coding DNA is just junk DNA with no purpose, but we're actually learning right now that that's actually not the case at all. So some types of non-coding DNA pad the ends of telomeres. Some make up RNA molecules that don't end up getting translated into proteins, for example. But a lot of our non-coding DNA regulates transcription of coding regions, which is what the scope of this project will be on. So these are called cis regulatory elements. From now on in the presentation, I'll just call them CREs. And the cis means that these elements are found in the same strain as the gene that they regulate. And here's a very general figure of examples of CREs. So promoters and enhancers are both examples of them. Some other examples are silencers you might have heard of. So promoters and enhancers both encourage gene transcription, but promoters are found right before their gene typically, while enhancers can pretty much be located anywhere on the DNA strand. So when certain proteins like transcription factors bind to the enhancers or promoters, then transcription gets initiated for the gene. So we're just starting to figure out CREs. There's a very big gap in knowledge in knowing which genes CREs regulate. And unlike this figure, it's extremely rare that CREs are just paired to a single gene. They often regulate many, and it's a really complex web that we don't fully understand yet. So my project and my research lab is specifically focused on the genomic evolution of CREs. And my next slide will show more of a visual of these questions. But recently, it's been very accepted in the scientific community that a lot of unique human phenotypes are actually a result of evolutionary changes in regulation, not necessarily mutations in our coding genes. So a previous project in our lab found that of documented CREs, they found which ones are unique to us evolutionarily and which are functional and active in the human brain. So my project is building off of these CREs to see if they regulate known aging genes in humans. So here's an overview of the data set I started off with based off the previous project in the Babbitt lab. So using statistical techniques, they found 722 CREs that have been evolutionarily selected for in recent human evolution. And because they've been conserved among vertebrates for billions of years, they're assumed to be functional. But in the recent evolution compared to other primates, they've had significant positive selection. And then also 179 CREs were found to have high activity in human brain neurons and induced pluripotent neural stem cells using an assay that's called lenti-MPRA. And the overlap of these two categories only had 34 CREs. So by researching these two categories, we can understand which active and brain-related regulatory regions have been selected for throughout evolution. And so what I was curious in is which of these CREs regulate known aging genes. So humans have many unique aging phenotypes when compared to other primates. So I was curious whether the selected and active regulatory regions have links to these genes that are known to regulate human aging. So there's no need to read and learn each one of these, but these are the 64 human aging genes that I picked out based on a very in-depth literature review. So many of these function in cell longevity, but some are a little different. Some maintain telomeres. Some are tied to aging-related cognitive decline or dementia based on GWAS. And so using genomic databases and with the help of RStudio, I found which of these 64 aging genes are regulated by any of the CREs that were described in that previous slide. So of those 64 original genes, 6 were regulated by either selected or differentially expressed CREs. In the case of CDKAL1, it was actually experienced both of those properties. So it's important to note that this is based off of known and documented CREs. There are likely many more out there that have not been annotated enough yet to have the data out there. But based on this data, these theories are very important. It's not shocking that the aging genes AKT3, CDH13, and WOX are regulated by CREs with high brain activity. But what's interesting is that there's now evidence that CSMD1 and UNC5D are regulated by CREs that were positively selected for. And CDKL1 is particularly interesting because it's regulated by CREs that have both of those categories. And so this makes that gene and its regulatory elements a very promising candidate for importance in our unique aging processes of our brain. And this figure shows the distance between those aging genes and their CREs. This is calculated using data from the UCSC genome browser. And the reason there's more than 6 counts is because each CRE and each gene have more than one region that it spans. So these aging genes are more often regulated by CREs that are closer in distance in terms of base pair count, which could suggest that some of these CREs are promoters instead of enhancers. But the distribution isn't that extreme, so regulation is still coming from CREs that are quite distal, as you can see on the right, up to 2 million base pairs away from its gene. So this slide and the next one, I haven't gotten to the analysis yet. So sorry for the slide. Obviously in the final draft, it'll be completed, but I just don't have enough info to speak on it right now. So in summary, specific CREs that regulate aging related genes have been identified as either CREs that are selected for or CREs with high brain activity or even both. So this gives us a more in-depth understanding of what their causal roles are in human neural phenotypes. More broadly, we can take another step towards understanding what genomically makes us human and how our brains develop and age over our lives. More studies definitely need to be conducted in order to verify findings and investigate other genes. Excuse me. In addition, it's very important to note that the category of select CREs is based on chosen parameters. So if using slightly different parameters, other CREs could also be identified as being evolutionarily important. And finally, aging is a major risk factor for development of neurodegenerative disorders like Alzheimer's disease. So in the future, regulatory roles in these diseases should definitely be further investigated. Thank you very much for listening. Let me know if you have any questions. Thank you.